Hello, and welcome back to another episode of Biology Explained with Mr. Hill. In today's episode, we are going to be looking at proteins, specifically looking at enzymes, their structure, how they work, and indeed how the rates of enzymatic reactions are affected by things like concentration, temperature, and pH. Now, here in Scotland, you will find this topic in Unit 1, Key Area 4, but you will also find that this video is relevant to GCSE and indeed many other biology courses around the world. Now, if you like this video, please do hit subscribe, like, and do share it with your friends so that they can also improve their biology knowledge. Now, let's make a start by looking at the types of proteins we find in the human body. Welcome to the first part of this episode, where we're going to start by introducing proteins and what types of proteins you will find in different organisms. So I'm going to mind map, as you can see on the screen here, the different types of proteins that you can find inside the body. Now, proteins, as you know from lower down the school, are important in our diet for growth and repair. And if you dig a little bit deeper as to what growth and repair really means, it means building cells and having well-functioning cells. And so part of that function involves proteins that do lots of different things. Some of them have sort of scaffolding roles that allow tissues to be built upon it. Others have more interesting roles where they are involved in chemical reactions. But all of these are proteins, and so without these, our cells will not function, and so our tissues, organs, and then ultimately ourselves will not function correctly at all. So, on our mind map then, we're going to begin by looking at the first type of protein, and that is the one that we're going to study a lot more on this episode, and that is the humble enzyme. Now, an enzyme is a biological catalyst. It speeds up reactions, and we're going to look at that in the next couple of parts in this episode today. So I'm not going to say too much more for now, only that they are a really large family of proteins that have a really important job in making sure cells can function properly. Next, we have antibodies. Now, you may have heard of antibodies before. Antibodies are produced by lymphocytes, which are white blood cells. So that links into some of our Unit 2 topics, which we'll look at later. And these antibodies are responsible for immunity, and they are produced by these white blood cells, and they bind to the antigens of pathogens, of disease-causing microorganisms. And by doing that, they flag them up to be phagocytosed by other white blood cells, and they also stop these pathogens from moving around or from invading cells, and so they're really important to keep the body safe from infection. Next, we have another large class of proteins, and they are known as hormones. Now, hormones are produced by cells in order to change the behavior of other cells. They are responsible for signaling within our bodies and for communication between different cells and different tissues. Again, there's going to be a whole episode on hormones for Unit 2, but they are a protein, and they're extremely important as well, so we need to mention them in Unit 1, 2. The next type of protein is one specific one we're going to mention is hemoglobin. Hemoglobin. Now, hemoglobin is found in red blood cells, and it's responsible for binding to oxygen to allow oxygen to be delivered around the circulatory system and to all of our tissues in our body. Now, it is a globular protein. All of these four so far are globular proteins or functional proteins, in so much that they have specific, quite interesting jobs to do. And they have a 3D kind of globular shape. And hemoglobin is one of those specific ones that have a specific job, and that is to bind to oxygen and to help deliver that around the body. Again, in Unit 2, we look at blood components in a lot more detail. And we'll return to this idea of hemoglobin and how the red blood cell is adapted to its job. Next, we have receptors. Now, we've already looked at this in our episode on cell membranes and transport. I'll post a link up somewhere here if you need to go back to have a look at that. Now, these proteins are found within our cell membrane, and they are responsible for picking up hormones so that the cell then changes its behavior. But they're also responsible as well for allowing molecules in and out of the cell. Okay, so the cell membrane, if you remember, has proteins embedded in them or crossing through them. and they're responsible for receiving information or for allowing molecules to enter and leave the cell. The last type of protein are structural proteins. Okay, all of these before are functional proteins. They have specific functions. They're quite interesting functions. The other family of proteins are structural. Now, they have a specific job too, but it's slightly less interesting. They're more like sort of scaffold, if you like, for cells and tissues to be supported. Okay, so we're thinking things like collagen, which you might find in skin or in cartilage, which helps to give the strength to the tissue. You're also thinking of keratin, which we find in hair and in nails. Again, it's there to support or to protect or to add structure to an organism. Okay, so here are then our 6 types of protein which you need to be able to name for your National 5. We have enzymes, antibodies, hormones, hemoglobin receptors, all being functional proteins, all with specific jobs to do. And we have a family of structural proteins such as collagen or keratin that are there to keep your cells and tissues strong and supported. Okay, so in our next section, we're going to look at protein structure in a little bit more detail. So we'll see you very soon. So now that we've looked at the variety of different proteins that exist in our body and all the different functions that they have, it's time to now look at the chemical and biological structure of this very important molecule. Now on the screen, you can see that I have drawn 3 different stages of organization of the protein structure. And we're going to begin with this squiggly line over here. Now, when you look at a protein, the protein is made up of a string of different amino acids. And all the amino acids have slightly different chemical properties. Now, we don't need to go into much detail, or any detail really, for the National 5. But because they have different chemistries, they end up forming a 3-dimensional globular shape of the protein that is very specific to the protein's function. And it's very important that these amino acids are in that right sequence so that that shape is produced when you're making the protein at the ribosome. Now, if we look at the string here of protein at a much more sort of closer level, you will see that they are individual amino acids that built up this chain here. And so on this chain, I have these little circles representing different amino acids. And if you remember back from episode 3, we looked at protein synthesis. Each of these amino acids are built at the ribosome according to the RNA sequence that the ribosome is reading. And so for this example, we have a yellow amino acid being bound to a purple, being bound to the yellow again, to a green, a red, a blue, and then back to purple. And when you have this long chain of amino acids, it will start to fold and bend into this wonderful 3D structure because of the different chemistries of each of these amino acids. Now, amino acids themselves have this chemical structure. Now, you don't need to remember this structure for your National 5, but I'm putting it here because it gives you a little bit of context as to the actual amino acid itself. We've got different elements here, which you will have recognized, and one in particular that you do need to know about, if you're National 5, is nitrogen. Okay, so an amino acid always has an amine group here, which has nitrogen included. And to link to Unit 3, when we look at crop production and the nitrogen cycle, this explains why nitrogen is really important for plants, because plants need nitrogen as nitrates from the soil in order to build their own proteins so that they can grow and so that they can have all the functions that we just looked at in the previous section of this episode. So to recap then, amino acids have a certain chemical structure that includes nitrogen, and amino acids are bound together into a chain at the ribosome, which we looked at in episode 3. These amino acids form a chain, which then starts to fold into a certain and specific 3D shape due to the different chemistries of the amino acids. And this 3D shape then determines the function of the protein. Okay, so that's part 2. We have now looked at the structure of the protein. Now let's move on to look at one family of proteins in particular, and they are the enzymes. So for the rest of this episode, we are now going to be looking at one family of proteins in particular, and they are the enzymes. Now, enzymes have very important and quite diverse roles within biology. They are found everywhere, and without them, Chemical reactions just won't happen at the rates that they're needed for organisms to survive. So they are an extremely important biological molecule, and that's why we study them at the depth that we need to for our National 5. So let's make a short definition of, first of all, what an enzyme is. So an enzyme is a biological catalyst. And we use these words because, one, it's a biological molecule made by cells, and two, it's a catalyst, which speeds up reactions without them being used up themselves. So in chemistry, you will have looked at catalysts as chemical catalysts, and biological catalysts are pretty much the same, just that the molecules are made in a biological context. So they speed up reactions, and they don't get used up. And they're the 2 points of a catalyst. Speed up reactions, and they are not used up. Now, enzymes have a very specific shape. And you can see on the screen here, I've drawn an enzyme here in purple, which has a sort of funny sort of a jigsaw puzzle shape cut into it. This is known as the active site. And the active site is where all the magic happens in an enzyme. Think back to our last section when we looked at that 3D shape of the protein. The shape is really important because you'll end up, for an enzyme, having a certain part of it where the reaction can actually occur. And that shape is really specific. And so if there was any change to the shape at all, then the enzyme won't work. So let's make a note of this then. Enzymes have an active site. And this is where the reactions occur. And the shape is also specific to the substance that it is either breaking up or building up. So the shape is specific. It's specific and it's complementary to the substrate, and that is the substance that it is working on. So the shape is specific and complementary to the substrate. So let's have a look at our diagram, and I'm going to add a little bit of steps as we go through. So step 1 here, we have our enzyme, and it has its active site. And I'm going to draw now the substrate that it works on, and the substrate will look a little bit like this. As you can see, it fits neatly into the shape of the active site. So this is our substrate. Now, just to reiterate this, if we had a different substrate, that shape maybe looks like this, okay? It will not fit into this active site. So this enzyme cannot work on this substrate. It will only work on the blue one here, which is specific. Now, the substrate then is going to enter the active site. So if I just draw it here, I'll color this in blue, you'll see it fits nice and snugly into our active site. And what is formed is an enzyme substrate complex. Okay? So the enzyme and the substrate, they lock together, and this whole thing is known as an enzyme substrate complex. Now, the enzyme will work as magic. The reaction will happen here, and the substrate will then be broken down into its different products. So, for example, I might have a new product here and a new product here. And let me color these in differently so we know that they're products. Let's do green. So I've got products here and a product here. Okay, so these are our products, and the products are free to leave the enzyme substrate complex, and the enzyme is then free again to work on its next substrate. Remember, it is a catalyst, so it's not getting used up. It will find a new substrate and it will keep working. Okay, now these steps, step 1, step 2, and step 3, is known as the lock and key hypothesis. Just like a key will fit into a lock perfectly, and only one key will fit into one lock, it's the same way that only one substrate will fit into the active site and work its magic. So that's how an enzyme works. They're biological catalysts, and the lock and key hypothesis explains their function. Now we're going to be looking at different examples of enzymes and the types of reactions that they catalyze. So now that we've explored how enzymes work and the process of their operation, now let's have a look at two different types of enzymatic reactions. One is degradation, and the other here is synthesis. Now, one of these is going to break a substrate down into individual little products, and the other is going to build up a new product from smaller substrates. Let's start with the degradation reactions. Now, degradation reactions are those that are going to break things down. So these are going to break substrates down. Okay, so there are many examples, but there's a couple that you need to know for your National 5. And that one, the first one, is going to be amylase. So amylase is a degradation enzyme which we find in our digestive system, in our salivary glands, but also in our small intestine, which is going to take starch, which is a complex carbohydrate, a long-chain carbohydrate, and it's going to break it down into glucose, the individual units of starch. That enzyme is amylase. Okay, now we know when amylase is working well in an experiment because we can test the solution that you've added amylase to for starch using iodine. Now, starch turns iodine blue-black. So let's just make a note of that. Turns iodine blue-black. So after a certain amount of time, you'll expect the amylase to have converted all the starch to glucose. And so when you add iodine at a later stage and it stays brown, then you know that the amylase has worked. And this is a traditional experiment that we do in school. I'm sure you've done this with your teachers. And it is also a good question that comes up in exams, because then they can start to ask you not just about the science of enzymes, but also how you would develop a valid experiment, or to do some calculations on percentage change or time for reactions to occur. So it's a good practice to have a look at the starch to glucose experiments using iodine. Remember, iodine turns blue-black in the presence of starch, and you can use that as a measure of how fast an amylase reaction is occurring. Now, if we look at synthesis reactions, they're the opposite. So they're going to build a product up from smaller substrates. So this is going to build products up. And we can look at the enzyme that does the opposite to amylase. So to build starch, we can build it from glucose, or strictly speaking, glucose 1-phosphate. So that's a 1-phosphate. And this is going to be built up to starch using the enzyme phosphorylase. Okay, and this is the opposite way to amylase. Okay, so glucose 1-phosphates will be stitched together using this enzyme to make starch. And again, you could use iodine to see if your reaction has been successful. So imagine, for example, you might mix phosphorylase with glucose 1-phosphate and start a timer, and every minute take a sample and test it with iodine. And when that iodine goes blue-black, you know that you have started to make starch, and that glucose has successfully been stitched together. You can then play around with some independent variables. For example, you could look at the effect of temperature or on concentration or pH on this enzyme, and you can then measure how fast this reaction has occurred. So, types of enzymatic reactions then. Degradation reactions break things down. Synthesis reactions build things up. And we've got amylase and phosphorylase as our two examples. In our next section, we are going to look at the rate of enzymatic reactions and how different factors affect how fast these occur. So now that we've looked at how enzymes work and the different types of enzymatic reactions that exist, we're now going to have a look at what the different environmental factors might affect how fast these reactions occur, or how the rate of the reaction will change. Now, enzymes are biological, and so by their nature, they are going to be sensitive to a lot of environmental conditions, such as temperature and pH. The other thing that the rate will be dependent upon will be the concentration of the enzyme itself. And so what we're going to do over the next 3 mini sections is to look at the relationship between concentration, pH, and temperature on the rate of reaction. Now, we're going to do it by graph form, and we're going to be also looking at a couple of exam techniques here. We're going to be looking at how we will describe a graph as well as explain it. So let's make a start with concentration. So as you increase the concentration of enzyme or even substrate, you are going to increase the rate of reaction. And so we're going to have an increasing graph like so. However, at some point, you will come to saturation, where any further enzyme is just going to be hanging around. And so it will be in excess, and the rate will no longer increase. And so we see this flatlining, this plateauing occur in this graph. The point at which the rate no longer increases, even if you increase the concentration, is the saturation. Okay, so there are 2 stages then essentially to this graph, stage 1 and stage 2. And at stage 1, when we're describing the graph, we can say that an increase in concentration, and I'm going to just write in shorthand here, obviously write these in full sentences in an exam, but the increase in concentration is going to increase the rate of reaction. Okay. However, beyond saturation or above saturation, okay, any increase in concentration does not increase the rate anymore. Okay, the rate will plateau. And what we've just done there is describe the graph. Okay, we've just said what happens. Now, if I had some numbers here and you were asked to describe a graph, you would maybe throw in a couple of numbers. So at what point is saturation in terms of concentration? But all we've done is saying what happens, and that's an important exam technique. If you start saying why, then you're not describing anymore, you're explaining. And if you're asked to explain, you've got to make sure that you don't just say what's happening, but you say why. And that's what we're going to do now. So it's quite simple, really. In section 1, the reason why the rate increases because concentration increases is simply the fact that there is more collisions between enzyme and substrate. So an increase in concentration is going to equal an increased chance of collision, which is going to increase rate. Okay? You've got all those molecules bouncing around at random, and if there's more enzymes there, the chances are that are going to hit a substrate more quickly than if the concentration was lower. Beyond saturation, though, what you end up having is excess enzymes, okay? The more you put in, there's not going to be any kind of more substrate for them to work on than before. And so at that point, there's a lot of enzymes hanging around not really doing anything. So above or beyond saturation, excess enzymes are not being used. And so the rate will no longer increase. And so what we've done there is that we have explained the graph. Okay, so whenever you see a graph in any exam question, okay, if you're asked to describe it, just say what's happening. Tell me a story as if you're moving from 0 all the way up the graph. And if you're asked to explain it, then you've got to say why and link that to what's happening. Okay, so it's typically an explain might be worth a couple more points than a describe. Okay, so that's the relationship then between concentration and rate. In our next mini section, we're going to look at the relationship between temperature and rate. Now that we've looked at the effect of concentration on the rate of reaction, it's time to turn our attention to the effect of temperature on the rate of reaction. Now, the temperature curve is much different to the concentration curve, and there are specific reasons for that. So let's start by drawing out the relationship between temperature and the rate of reaction. Now, as you might expect, as you increase the temperature, you are going to increase the rate of reaction. And this is the case when the temperature is low. Now, at some point, you are going to hit the enzyme's optimum temperature, i.e. the temperature at which the enzyme works its best at. Above that temperature, the rate is going to crash, and it's going to come back down to 0. And we can label our graph even further with our optimum, which is there. Optimum. Now, we're going to do like we did before. We're going to describe and we're going to explain the graph. So we're going to start by describing, and there are 2 sections to this graph, section 1 and section 2, before and after the optimum. So let's start by describing these 2 stages. So let's start with this first stage. Again, as you increase the temperature, and I'm going to write this in shorthand, but you can write in full sentences. When you increase the temperature, you're going to increase the rate of reaction up to the optimum. Okay, so if the optimum was 40 degrees and you had 20 degrees and you increased it to 30, then you're going to increase the rate of reaction. Above that, though, any increase of temperature above the optimum is going to result in a decrease in the rate of reaction. Okay. And so what we've done there is we have described the graph. Okay, this is our description only. Okay, if you're asked to describe the graph, that's what you do. As you increase the temperature, you increase the rate of reaction up into the optimum. Any further increase in temperature will decrease the rate of reaction. Now, for an explain question, you need to go into a little bit more detail, of course, as to why this is the case. So let's start with the first section. As you increase the temperature, you are going to be increasing the kinetic energy of the enzyme and the substrate. Now, think back to your first and second year science. You will recall that all particles have a kinetic energy, and this relates to how those particles are moving and vibrating. And when you increase the temperature of a substance, the particles will start to vibrate more. And that's exactly what's happening with our enzymes and our substrates. When you increase the kinetic energy, you are increasing the movement of the molecules, and so you're going to increase the chance of them colliding and thus increase the rate of reaction. So it's going to increase movement, increase chance of collision, which equals an increased rate. Always link it back to the question, okay? So you end your sentence with a therefore, this increases the rate of reaction. Now, beyond the optimum, things get a little bit more interesting. When you increase the kinetic energy of a enzyme above its optimum, what you end up doing is you end up causing the enzyme to vibrate just that little bit too much, so that the active site and the 3D shape of that molecule begins to alter. And when the active site changes shape, the substrate no longer fits in snugly, and so the reaction no longer occurs. And this change is permanent, and so the enzyme is no longer functionable, and the rate, therefore, as a total, will decrease. We called the process by which an enzyme's active site changes shape permanently and the enzyme losing its function, we call that process denaturing, and so the enzyme becomes denatured. So let's now write this down as our explanation to step 2. So any increase in temperature above optimum equals a change in the shape of the active site, and this equals a decrease in the rate as enzyme is denatured. Let me write that out for you there. Now, very important point, enzymes do not die at high temperatures. That's because they were never living in the first place. Remember, we are talking about molecules, not about cells, so we can't say they die. Instead, we use the term denature. The active site has been vibrating so much that it just changes shape and the substrate no longer fits in it. And so there we have it. The explanation to why temperature changes the rate in the way that it does. And again, if you are asked to explain a graph, you need to say more than just what's happening. you've got to tell me why. And typically this will be 2 to 4 marks for your question. So last but not least, we're going to now look at the effect of pH on the rate of an enzyme reaction. This time, though, we are going to start our graph somewhere in the middle, where the enzyme is working at its optimum. So, for example, I'm going to start there. Now, with the pH relationship, we have an enzyme working at its best at a certain pH. And this is different in different enzymes. That's because enzymes find themselves in different conditions. And so the optimum will be different for each of them. So, for example, pepsin is a protease, so these break down proteins, and they are found in the stomach, and so have to exist in stomach acid. Therefore, pepsins are going to have a lower pH optimum than an enzyme that's found somewhere else in the body. Now, whenever you increase or decrease the pH away from the optimum pH, the rate is going to decrease. And so what you end up having is this sort of bell-shaped curve. So if your pH was to reduce, the rate will reduce. And if the pH is going to increase, the rate will also reduce away from the optimum. And this optimum is specific to the certain enzyme. Now, they're not really going to have part 1 and part 2 in this graph. Instead, we're just going to just have a general point here and say that each enzyme has a specific optimum pH related to its environment. And that is basically I'll describe and explain. We don't need to go into too much detail really in this. We can say, though, that enzymes don't really get denatured when the pH becomes higher or lower, unless they go to the extremes. For example, if this was pH 7 and you had the enzyme working at pH 8 and then back into 7, the rate should increase because the changes to the active site are only temporary when the pH changes. Unless, of course, you go to the extremes, in which case you might cause some denaturing. So there's no real denaturing when it comes to pH changes. There are, though, of course, with temperature. Okay, so there we have it. We've covered then the effects of 3 different conditions on the rate of reaction. And again, as I said before, it is quite likely that you'll face a question about this in the context of an experiment. So be prepared to maybe either complete a table, complete a graph with some data, as well as describe and explaining what you see in that graph. Okay, so it's question time, and today's question comes from paper 2 from the 2022 Biology N5 paper, question 3. And this, as predicted, is a question related to an experiment and enzymes. So give it a read. This will take you probably about 6 to 7 minutes to complete. So pause the video, and when you're ready, come back and we will go through the answers. Best of luck. Okay, let's now see how well you have done. So let's go through the first question here. Let me change my pen to a green pen. First of all, an investigation was carried out into the breakdown of cooked egg white by enzymes. 2 grams of cooked egg white was placed in each of the 4 test tubes containing different enzyme solutions at different pH levels as shown. So A and B are both pepsin, and C and D are catalase. A and C are pH 2, and B and D are pH 7. The test tubes were placed in a water bath at 37 degrees for 2 hours. The cooked egg white was then removed and weighed. The results are shown in the table. So we've got the mass after 2 hours, the change in mass, and the percentage change in mass. And you can see that there's a gap here for A. B has been done for you, and C and D didn't change mass at all. So, question 1. Complete the table by calculating the change in mass of the egg white in test tube A. Okay, so this is quite simple. We know that it started off at 2 grams at the start, and it's now 0.3 grams. So it's just 2 minus 0.3. It means it's changed mass by 1.7 grams. Okay, so it's really important for your exam questions that you read the questions carefully. Okay, sometimes the information and data is in the introduction blurb before the question comes in. So don't speed through it. Make sure you're taking your time and you're picking out the data that's there. Okay, next question. Explain why the change in mass in test tube B was less than that in test tube A. Well, we know from looking at the rates of reaction that enzymes have a specific pH that work at their optimum. And so clearly the optimum for pepsin must be closer to pH 2 than it is pH 7 because it didn't change mass much at all. The egg was not really digested at all by this enzyme. And so your answer for this question is going to be that pH 7 in test tube B is not at the optimum. Or any words to that effect, where you say that pH 7 or test tube B was not working at the optimum pH for the enzyme. Question B2. Explain why there was no change in mass in test tube C and D. Now, catalase is an enzyme, it's a degradation reaction enzyme that breaks hydrogen peroxide down to oxygen and water. And so clearly there is no hydrogen peroxide in our enzyme, in our egg white, for this enzyme to work on. So there's not going to be a change in mass. Okay? So you can just say simply that catalase does not break down. You could say digest egg white. Okay? And if you know that hydrogen peroxide is a substrate, if you said hydrogen peroxide is not present there, then that will get you a mark as well. If the investigation was repeated at 70 degrees C, describe the changes in the structure of the active site of the enzymes. Okay, so we're just describing here. So the active site will change. Okay, so because we've increased the temperature beyond clearly the optimum here. And so you can say that it will change, it will become denatured. Okay, and then finally describe the change in the rate of reaction in test tubes A and B. Well, the rate of reaction in a denatured enzyme will reduce. So it will reduce. Okay? Or if you say it became 0, or the rate of reaction will decline, any of those sort of, points will get you the mark. Okay, well, I hope you've done well in those answers. As you can see, quite a lot of the time you're going to be faced with a little bit of maths and a little bit of describing and explaining of the rates of enzyme reactions. So there we have it. We've covered a lot today, from protein structure right through to how the enzyme reactions are affected by things like temperature, concentration, and pH. Now, this is a large topic in Unit 1. It's very likely you'll be facing some scientific literacy questions or some graph questions on this topic. So do practice your math skills, do practice your graph skills, and remember some of those key words that we've used today. Things like denatured, saturation, and optimum. As always, if you liked this video, please do subscribe, and we look forward to seeing you in our next episode, where we'll be looking at genetic engineering and how we can use GM crops and GM bacteria for good. See you now. Bye-bye.